Welcome, attendee, to the 2025 Bioinformatics lecture of the Biophysics series. So given by Stephen Wingett and myself, Tim Stevens. Now, an hour for bioinformatics is just a bit silly, really. So I'm just going to give a light overview, specifically for biologists. I'm going to do the sort of protein-y bits, and Stephen's going to do the sort of DNA bits. So it's just a brief overview with tips. I've included tips in red, and they sort of take-home messages that might help you overcome a few difficulties. We take the wider view of bioinformatics as being this broad intersection between computing and biology, with some math sometimes, especially to do with statistics. Traditionally, bioinformatics, as people think of it, as just sort of sequences. That's kind of how it started, of course. We only needed computers to deal with big things like DNA and protein sequences. But these days, it encompasses a wider array of organisms and a wider array of molecules. From just protein, everything contributes to biological function in the end. And then you're adding more dimensionality, structures, interactions, and bioinformatics covers all of these, including, higher order things to do with the genome, chromosome folding, epigenetic regulation, that sort of thing. So this is the context that we set ourselves in. And if you want to get started in bioinformatics, it really sort of depends on how much commitment you can put in. Most people will be happy to dabble as a biologist and not really do programming or anything confusing like that. There are loads of web tools out there these days. When I started, there were very few. You had to do it all yourself. But if you're doing simple stuff like alignments, finding orthologs, whatever it is, there's an established technique, there's likely to be a web tool. We list quite a few of them in the talk, so this whole talk, or the slides, can act as a reference for you. We tried to keep it relatively up to date. And there's lots of people like Stephen and myself in the building. There's Tom in proteomics, Alistair in PNAC, and various others. There are friendly bioinformaticians that can help you understand what's going on, help you use particular pieces of software, even help you with coding. And we're particularly keen to get people started on the right course in the first instance. So we like it when people come to us for experimental planning, particularly with sequencing. When you've got options like, do I use paired end, single end, longer reads, shorter reads, etc., we can advise on what's going to give you the best results, at least in the statistical sense, in the long run. We often get people come in at the last minute before they want to submit, and we think, well, why didn't you do another replicate or whatever it is? However, if you're really keen, and if you have a project in mind, and you've got some focus and have the data, you might wish to actually start tinkering with a computer. And we recommend the into that is to go to sort of courses and tutorials on command line computing, so you can use the cluster, so you can use your Mac terminals or Linux, and try some simple programming. And Stephen and I both offer courses on that. Just briefly, if you want to start a bit of computing for biology, we recommend trying Python first. You can use R as well for statistics, but I think Python is the best one to learn on, and it's the most popular language these days in the whole world. And if you want speed, you can get to compiled languages. And we can advise on all the development interfaces. We prefer Microsoft's VS Code at the moment. And we can offer a Jupyter service if you want to sort of do coding through a web page. And as mentioned, we offer courses on getting started. Okay, so it'll be back to me in a bit. Stephen's going to have a little chat to you about DNA and RNA stuff.
Okay, so I thought I'd start this talk by briefly discussing, you can hear me, yeah? Some of the tools that are available to you in the form of online databases. So if you think bioinformatics can be thought of the intersection between biology and the analysis of data, and there are already a multiplicity of databases available to assist you with bioinformatics and your research. So the number of resources available is increasing at a rate that most people find hard to keep up to speed with, and I have to say I'm one of them. And the content of these sites is not only increasing in terms of the volume of the data being deposited, but also in the different types of data that's being deposited as well. So it's becoming sort of ever more difficult task to keep track of this. So just as an example here, this is a single UniProt. This is just a single UniProt entry. So UniProt is a proteins database. And you can see for this entry, there's links to a whole range of other databases, proteomics databases, gel databases, gene expression databases. So I'm just saying there's this huge ecosystem of data out there, and it's just hard to navigate what it is, where you get the information that you're interested in. So that's what I'm trying to do in the next couple of slides, just point you to the major resources in bioinformatics that people use. Also, if you want to go a little bit deeper, there was a paper in Nucleic Acids Research last year, which summarized, I think it was 180 resources. So a lot of them, I think about half of them were new and half of them were updates to existing resources. So that's also quite a more extensive place to look if you're interested in finding out the resources that are available. So I thought I would start by looking at the databases. And I was going to look at, start just talking about nucleic acid sequences and genomic databases. So Ensemble is probably the best known. It's maintained at the EBI, the European Bioinformatics Institute in Cambridge. And it began life in 1999, just before the first human genome draft assembly was put together. And the idea was to integrate this genome data with other biological data and make it publicly available, which they did. And since then, it's been expanded. More genomes have been added, more data types have been added, such as comparative genomics, variation data, gene regulatory data, and so on. And since then as well, there's been, like 2009, there's been spin-out sites where you can get data just exclusively on bacterial genomes, protist genomes, plant genomes, and so on. So it's been continually expanding. And I think in 2020, it reported over 50,000 genomes. So it's a huge genomic resource. But fortunately, it is well maintained, it's well documented, and Hinkston even provides training courses where you can go and learn about it. So that's like a primary resource for genomes. The rival, I guess, if you think of it in those terms, is the NCBI, which is the National Center for Biotechnology Information, and that's based in the US. Ensemble, as I've mentioned, is in Europe. They're very similar. There's a lot of overlap between the two in terms of the data they hold. Maybe you could argue that Ensemble is a little bit broader, NCBI is a little bit broader, Ensemble is a little bit more human focused, but essentially there's a lot of overlap between the two. There's also UCSC, that's University of California in Santa Cruz, is a genome database. It's very good again. It scores particularly well in the field of comparative genomics. So something like the liftover tool is available there, where you can perform different, well, it's a tool where you can convert coordinates between one genome assembly and another. So yeah, so there, if you had to, sometimes people ask which one should I choose. Normally I pick one that, normally if I'm on a project and I've been using one genome assembly, I tend to stick with the same genome assembly. So I've been working with Ensemble, I tend to stick with that. Often it can just come down to which one presents the data the best. And most easily accessible. So that's genome databases. There are also gene databases as well. So GeneBank contains gene information. It also contains information on different types of sequences, such as express sequence tags and other types of sequences, or maybe even genome assemblies before they're ready for publication. So it's a sort of miscellany of different molecules are stored on GenBank. It's a bit of a wild west, but it is very large, and often you can find what you want there. If you want something a bit more curated in a similar vein, RNA-seq has automated and manually curated genes on it as well, and it's also non-redundant in its nature. So it's a sort of more curated, more structured repository. Sorry, so UniVec, when we data deposition for vectors, you could go to UniVec. This is a good resource. So it has vectors, it has primers, linkers, that sort of molecule. You may be wondering why you'd be interested in that. I mean, you may be interested in that, but often vectors form a source of contamination. So if you can map against that, you can maybe try and identify why your experiment's not looking as it should do. Sometimes when people are performing mapping, they're mapping against what they know, but they don't think about what it is that could actually be there. So UniVec is a good resource. Genome browsers, I guess most people are familiar with this idea. Basically, it's a software tool where you can visualize and explore genomic data. So this is a screenshot of one you're probably familiar with. The idea, it presents a user-friendly graphical format where you can see features like gene locations, splice sites and so on. And the idea is you can use the mouse pointer to zoom in and out of specific regions. So it's essentially like a digital map of the genome. Ensemble produces a very good genome browser, as does UCSC. WashU is also very good, the Washington University genome browser. I tend to like that. Sort of in the past, I've used it because it's very good with epigenomic data. So I've displayed like high-C interaction arcs on there. It's very good for that sort of thing. If you're working on non-model organisms, the sort of satellite sites like Flybase, Yeastbase, they can have their own genome browsers for their sort of more niche organisms. So that can be quite useful as well. Okay. Oh yeah, I should mention Biomart. So Biomart's managed by Ensemble. In spite of its name Mart, it is open source and free. And it's essentially a place where you go shopping for data. So you sort of go in, you specify the database that you're interested in, the type of data you want back. So it could be gene data and the sort of format that you want your data presented in. And then you can go in and download it. So an example might be, you might be interested in some class of human genes that are of particular interest to you. You specify the human database, the genes that you're interested in, and then the sort of attributes you want back. So you might be interested in like coding regions, protein sequences, and so on. So that's essentially what Biomart can do. It's a huge resource for downloading genomic information. Okay. Taking longer than I thought. So expression and epigenomics, I guess people know what expression and epigenomics is. A major resource for this sort of data is the Gene Expression Omnibus, or GEO, as people tend to call it, and that's maintained by the NCBI. It stores microarray data, next generation sequencing data, and it provides a nice web interface where people can download data sets. So traditionally, I guess when you look at papers, you often see some sort of accession number when people have done some sort of sequencing experiment. And then more often than not, we'll refer to a GEO database. So the idea is that people have performed an experiment and they've uploaded their data to GEO and you can download it. It's worth knowing now that most journals now require, as part of the publication process, that if you've done a sequencing experiment, you do submit your data on a public repository. So and I would recommend GEO. I find the submission process there very simple and straightforward. So that's the Gene Expression Omnibus. ENCODE, it was around about, it started work, I guess, over a decade ago now, although it doesn't seem like it. It's essentially an international consortium to produce an encyclopedia of DNA elements. And it sort of builds, it's like a comprehensive list of functional regulatory elements in the human genome that control protein expression, gene expression, and so on. It's a very nice resource. It's very well presented. The data is sort of laid out in a very sort of accessible format. And you can get sort of studies there, such as RNA protein interaction studies, RNA expression studies in different tissues and that sort of thing. So it gives you a nice overview of regulatory genomics. Maybe just move a little bit more quickly through these. We've got transcription databases. So there's the EBI Expression Atlas, and that sort of aggregates public microarray data and RNA-seq data, and it looks at expression of these genes in different tissues and in different biological conditions. And then there's also BG, which looks at the expression of genes in different species, which is another useful resource. In a different vein, there is DETMAP, and this looks at expression of, well, essentially they did like a knockout study where they knocked out genes in different cancer cell lines. And what they tried to do was identify genes which were essential for the continued proliferation of those cell lines. So this is an interesting resource, and it might give an overview, an idea of what genes are essential for the continuation of cancer. Maybe if like they're related genes, like metabolic genes, it might indicate new pathways as they're essential for cancer maintenance, and maybe potentially give a sort of idea of potential new therapeutic targets. So that is the DETMAP. Then there's proteomics databases. So I'm Sorry, I'm bombarding you with lots of terms. I'll move on. There's the PRI database, which looks at the proteome of multiple species. And then, of course, there's the Human Protein Atlas, which is an exhaustive list of protein expression. And then if you want more standardized data sets, you can look at PAXDB. So that's just a list of the databases there. So just looking at you're all familiar with the idea of DNA sequence alignment. This is the idea of aligning sequences to one another, so 2 or more sequences to one another. But the type of aligner that you want to use will depend on the particular type of question you're trying to ask. So you may be performing some sort of whole genome alignment where you want to compare one genome with another. And for that, I would recommend tools such as PCAN or Multi-Z or Multi-Z. They tend to be good, but they tend to be quite sort of labor intensive, and they're quite slow to compute. So if you can get hold of a pre-computed sort of comparative genome, that is a good thing to do. So I was looking quite recently, there's a tool called Cactus, where people can align multiple genomes, and that's a nice resource online as well. If you're comparing a small number of sequences, for example, if you've got like peptide sequences and you want to identify consensus sequences and so on, you could use tools such as Clustal W or Clustal Omega. Or if you want to, maybe you've got a sequence that you want to check against a whole range of different species, you could go online and use BLAST or something similar to that. So on the right-hand side are more sort of reads types of sequences you would get back from the next generation sequencing experiments. So typically off a Illumina machine, you'll get short reads to about 50 to 200 base pairs in length. So they're short, but the actual throughput is very high. So you'll tend to get maybe up to billions of reads on the biggest Illumina machines. And what you would do is you would tend to align those to a reference genome using a tool such as Bowtie2 or BWA. And that will work out from where in the genome these reads arose. For longer reads that you'll get like off a Oxford Nanopore MinION, I would tend to use a mapper such as Minimap2, which has become a bit of a de facto standard. If you're looking at RNA-seq data sets, you have to remember that some splicing will have taken place. So you might not be able to align the reads immediately back to the reference genome. So you'll need a spliced-aware aligner such as STAR or HiSat2. Or you could either look into something called pseudo mapping, which allows you to process data sets quickly and can get quantitative matrices at the end. And then finally, there's single cell data sets where you're doing RNA-seq, but at a single cell resolution. There's 2 different technologies used at the LMB at the moment I'm aware of, as 10x and PARSE. Both of those have their own proprietary aligners. So 10x has Cell Ranger, and PARSE has a tool called Split Pipe. They're actually proprietary, but they're built actually around STAR. So that's the idea there. I'll just try and go through this using your time a little.
We're on for 5 to 10 minutes, that's all right.
Okay. So I just wanted to talk some of the terminology that's involved in next generation sequencing experiments. Sometimes people just aren't familiar with the terms that are being used. So the idea, if you performed your experiments, you've got your molecules that you want to sequence, what you'll tend to do is perform some sort of size selection to get the size of molecules you need. So that might be sonication or some sort of digestion. And then what you will typically do is ligate some sort of sequencing adapters onto either end of the molecules. And this allows them to be sequenced by whatever sequencing machine you're using. You'll then perform the sequencing, and something called base calling will take place, where it will call the bases, the individual nucleotides in your molecules that you're sequencing. And typically these are accompanied with what are known as quality scores. So a quality score is an assessment of how reliable a particular base call is. So that's quite useful in the alignment steps later on. Then what would happen is you would take your file. So a file will be output by your sequencing machine. And you will then normally perform something called quality trimming, where you will remove reads which are of low quality, or sort of remove ends of reads which are of low quality. And normally you perform some sort of QC analysis just to make sure the sequencing has actually gone as you expect. The data looks at a global level as you would expect. Then typically you would take the reads, as I mentioned before, and map them against your reference genome. And then essentially it's more down to your experiment what you want to do with them. So you may want to allocate them to reads, like you might want to allocate reads to genes in RNA-seq experiments, or you might want to identify maybe peaks where you get local aggregation of reads against your reference genome. Typically people use Python scripts or R scripts to interrogate their data, or there are user-friendly tools such as ChIP-seq, where you can import your data and then use like a drag and drop GUI to interrogate your data. Just a little bit of terminology. There are different types of what's called paired end reads and single end reads. So if you imagine my arm is the molecule you want to sequence, a single end will be where you just sequence from one end, paired end will be you sequence from that end and the other end, and the reads may or may not read in the middle, meet in the middle. So that's the idea. Also, you need to decide in advance how much sequencing you need to do. So, for example, if you're doing an RNA-seq experiment and you're trying to look for differentially expressed genes, you want to make sure you've got enough reads back that you'll be able to identify these differentially expressed genes. Okay, so that's a sort of overview. I'll just talk briefly about some of the tools that are involved. They sound a little bit esoteric at first, but if you know what they do, then they're not really too complicated, or at least have a broad idea of what they do. FastQC gives you a global overview of when you performed your sequencing. Just sort of, it's a sort of quality control tool essentially. Does the data look as I expect, or have I got the right number of reads? Are they of the right quality? That sort of thing. Trimgalore or CutAdapt are common bioinformatics tools to trim reads to a length or remove reads which are of poor quality. So it just improves the quality of your data set. Also, when you performed all your analysis at the end of a bioinformatics pipeline, typically there's a tool called MultiQC which collates all your data and generates a nice, simple, easily interpretable report at the end. There is also tools, once you map your reads, to actually interrogate those data sets that you've generated. There's tools called SAMtools and Picard. So they are standard tools on that people have installed on bioinformatics machines to actually analyze the output files. And then if you want to aggregate your reads from these, if you want to aggregate your data into a more manageable format, you tend to use something called bed tools or HT-seq. So for example, HT-seq, if you're performing an RNA-seq experiment and you want to turn your file of just map reads into some sort of data matrix so you can see for each gene and each sample the expression levels, you would use a tool such as HT-seq, which is nice. And then if you're doing variant calling on your data sets, you could use tools called GATK or Freebase. So they're popular tools that people use. Just briefly mention file formats. Faster formats are how people store sequence data. So essentially, it's just a text format with all the nucleotide sequences or the amino acid sequence in there. And typically, there's just a header line at the top, just telling you what the sequence is. FASTQ files are what come off of sequences. Again, they're just text files with sequences, and it also has a quality score assessment in there. Once you performed your mapping, you'll get something called a SAM file back, or if you compress it, it will be known as a BAM file, or it could be in gram format. This just essentially stores the location of where each map reads against your reference genome. There are different file formats as well. There's file formats for mapping, comparing multiple chromosomes to one another, or when you're looking at genome browsers and you might see the little peaks, that you see those sort of like termite mounds on genome browsers that represent like attack-seq peaks. These are in this wiggle format here. There's variant call VCF files which store variant calls, and then genome annotations are stored in these GFF, GTF file formats. And also 3D structures are stored in this PDB format as well. So that's pretty much what I was saying. I'll just very briefly, I try to install a lot of commonly used pipelines on the computer cluster here at the LMB. So if you do want to sort of do some sort of RNA-seq, ChIP-seq analysis or an attack-seq analysis yourself, please do come and have a chat with me. I'll try and point you in the right direction, and then we can make the sort of initial processing quite smooth.
Yeah, it's all installed in the cluster, and it all works. It's all been shown.
Yeah, it should work. Okay. Thanks.
Thank you, Stephen. Okay, on from DNA RNA to proteins. Okay, and I'll skip through some of this. Okay, so Stephen talked about the DNA sequence databases. So it's worth mentioning the protein sequence databases. Like with the DNA data, there's a sort of European American split. So there's the NCBI protein database and the UniProt database, which comes from Europe. You take your pick of which you prefer the interface for. They basically cover all the same data. Just one word of warning: if you're used to NCBI data, particularly if you're using older publications, proteins were identified with a GI number. These are now obsolete. You have to map them to the latest IDs. UniProt IDs have issues, but they didn't completely overhaul like NCBI did. I prefer UniProt, and UniProt actually was an amalgamation of various other European databases, including SwissProt. But it still maintains the SwissProt, although that's an old database, it maintains the SwissProt label for the well-curated protein sequences that humans have actually looked at and checked. And so you'll find the most, cross-links, the most rich features actually in the SwissProt section. UniProt does have everything that's ever been translated automatically from a genome, and that's the TREMBL section. There's less annotation there, but it at least going to be complete. So those are the major databases. However, when you're sort of looking through for information in protein sequence, obviously you don't want to search the whole lot. So we're generally thinking about subsets of databases to look in. Obviously, you can look on a per species level. That's all we want to do often. And species are organized in terms of their proteome, so all of the expressed proteins in a given species. So you can see here, there's some Drosophila ones. UniProt is handy, and it gives you the level of completeness of the proteome as assessed by orthologs it reckons should be there. And this is using the buzzcode method, which you can sort of see up here. So you can find the most complete proteomes if a choice of species is available to you. Otherwise, if you want to search across multiple species, you can search all of UniProt. However, UniProt has a degree of redundancy, and it can be extreme sometimes. So here's a search I did for human cytochrome B, and it comes up with almost 2,000 entries. This is because it's a mitochondrial sequence. People have been sequencing this since we could sequence, and everybody wants their precious sequence still in the database. Nobody wants to remove anything. So there's an awful lot of redundancy. However, one sequence will be the reference sequence. So there's only one reference. There you go, in orange, cytochrome B. And so you can search for the reference. You can search in just the reference proteome for a given species. So you can do that. And there's also non-redundant databases. There's a vague non-redundancy with the UniPark. But the best non-redundant databases are the UniRef clusters. So what these databases do is they say, is there a degree of sequence identity, or it's identity, not similarity, identity between sequences at that 100%, 90%, 50% level. And so with UniRef 50, there'll be a cluster of sequences, and they will share at least 50% sequence identity with one other thing in the cluster. And then you have representatives, which are on the centers of clusters, or approximately near that. And this gives you a much smaller data set to search if your species are agnostic. So it allows for faster searches, and it still covers pretty much all of the sequence space. So go for UniRef if you're rather than search everything. It also reduces the number of hits you get back, which can be overwhelming sometimes. Okay, moving on, sort of further things to do with proteins. We're often concerned about proteins as families, so how they have evolved. I suspect you already know this, but that means we're concerned with proteins that arise through gene duplication, like paralogs, and how proteins diverge as species diverge, that is, orthologs. And I have 3 recommendations for sites you can go to help you, find orthologs and to visualize them. Tree family is my recommendation if you actually want to build pretty trees of how things are related. The best one in terms of information, in my view, is Panther. This is comprehensively, in terms of the species it covers, it's well maintained. It also has really good links to Go annotations. So that's gene ontology sort of tags which classify proteins and their genes with functions and sort of biochemical sort of information from experiments. And the idea with Panther using Go annotations is that you can spread the annotation, which they may be on Drosophila, right across all of the orthologs into all of insects and maybe into vertebrates too. So Panther is my favorite in terms of information. And if you do a gene ontology enrichment search from the Go website itself, it actually uses Panther orthologies to spread the information. So that's a great source of orthologs. Of course, proteins come in, at least in eukaryotes, as multi-domain globules, which are the units of folding and evolution. So we have a number of databases to do with those. One thing that's changed fairly recently is that PFAM has been absorbed into Pro. So there's now only sort of one big EBI, European-based database for domains. The protein domains are basically stored as multiple alignments expressed statistically, which is, we say it as an HMM, which is how the probability of one residue depends on what went before it. But this gives a way of classifying sequences we've never seen before, pretty automatically. You may be interested in domains that you have structural representation. So there's PDB-FAM there, which is just mapping PFAM back to the PDB. I came across this a few years ago, ProCite, whereas this is my reference for domains. I like ProCite though, because you can actually build these cartoons of your domain structure pretty easily on the website. That saves you a bit of faffing in graphics programs. Okay, so increasing the dimensionality here, there are various protein structure databases. Obviously, people mostly have heard of the PDB, which is split across, America, Europe, and actually there's a Japanese version as well. Remember that if you want the data that went into the calculation, if it's NMR, that's actually housed at the BMRB, which is actually affiliated with the PDB quite closely. So all the structures are in there, and it's very searchable. If you're interested in rather than a specific protein, but a specific shape or class of fold of protein, then I recommend the classification based on the structural characteristics. So there's Janet Thornton, others at London have the CATH database, and Scott came out of the LMB, Alexei Merzin and others. So you can search for proteins based on, has it got mostly allylic, beta sheet, is it, coil, that sort of thing. So if you're interested in structural class, that's the way in, I would say. Something I came across a few years ago is that, when manipulating these structures, I like to manipulate them programmatically. So for example, I can generate animations and, display lots of things in the same way for a publication. And discovered that UCSF Chimera, as well as PyMol, actually have lovely Python interfaces. You can script all your structure display. It's fairly straightforward, although annoyingly they're different in each case how you do it. It does work. These days, people tend to just go straight for AlphaFold if they want to see what a sequence is, and they completely forget or can't be bothered with comparative modeling. Now, bioinformaticians spent a lot of effort trying to make comparative modeling work really well, and it does work really well, but it only works as well as your sequences are close in some sense. And so if you've got a structure and you've got a protein, a homologous protein that is, sort of over 85% identical, comparative modeling may well be better than AlphaFold. And there's lots of tools out there to use. And the idea is that you take a known structure and you align your sequence of your homolog with that and identify the template to use. And then when they're aligned, that gives you the backbone, and then the side chains are added and you test how well the model works. So don't forget comparative modeling. If you've got a close homolog, it works very well. Okay, so I'm moving on to more sort of connection type things. Well, I've already mentioned the gene ontology. So proteins and also genes get labeled with tags saying what the proteins do in a biochemical sense. That's a molecular function. What pathway, what biological process they're involved in, what cellular component, organelle, etc. they're involved with. So these are annotations that come from experimentation. A lot of them are human, a lot of them are automatic these days, increasingly. The coverage can be patchy, which is why I recommend going through the Panther system and actually gene enrichment analysis, the enrichment analysis offered by the gene ontology itself actually covers whole orthologous families. I want to highlight, as well as the gene ontology, if you want to know about the function of proteins, I want to highlight these OMIM database, which is the online Mendelian inheritance in man. And there's also one for animals too, I think. And this is just a list of medically observed phenotypes, conditions, and relating them to polymorphisms, indels, structural changes in genes and proteins. Okay, and this is a resource that's been, been gathered over many, many years, and it's, got human beings doing it. It's high quality, and sometimes if something is not annotated in one of these sort of more informatics databases, then the medical database will say, oh, actually, there's a mutation in here which causes a syndrome because we've linked it. And so that's a really rich source of information that's often neglected in this in these days. Okay, function, of course, is not the function just of a single protein, but of all pathways and interactions between biochemicals and proteins and nucleic acids and whatnot. If you want to know about the existing pathways, then I will point you toward KEGG, which is one of the oldest ones. So if you want to know what glycolysis looks like, go to KEGG, it will have some pictures. It'll be thorough. It's a little bit dry, but it will be thorough. There's a more modern one called Reactor, which has these lovely cartoons. So again, you can cheat for, making pretty slides of your favorite, pathway, whatever it might be. And it will cover traditional biochemical processes like, glycolysis, citric acid cycle, whatever, but also sort of things to do with vesicles and cell division, etc. And it's very comprehensive and saves you a lot of effort in trying to work out what the components are, if it's known, of course. For the sort of lesser known stuff, we're reliant upon predictions of interactions between proteins. I say predictions that because although these databases are based on experimental data, these two hybrid, coevolution, or what have you, they're very noisy. There's a lot of false positives or contextual positives in here. So I treat these databases with a bit of caution. I was tempted to go to Intact as the main EBI one, but I've had it recommended to have a look at String, which is a little bit more straightforward. But, you can read, it's also BioGrid. So yeah, it's a vast network of interactions, only some of which will be important to you, but at least it cuts down some of the search space about what you could be working with. And actually currently myself and Stephen, to some degree, we're working on a new database for interactions, which should be a lot cleaner, based on lots of different experimental sources rather than just one, using neural networks, I hasten to add. Okay, now a little advert for the Unknown project. So this is something I was doing with Sean Monroe and Matthew Freeman. And this is, well, it's sort of like the opposite of the gene ontology. So we've got genes that we know about, and we have all these functional sort of labels assigned to them. Well, what genes have got no functions assigned to them, but are also important because they are conserved? And so this is an analysis that we did just to show that if you look at proteins that are conserved between humans and fruit fly, you will assume they're important. We found that 10 to 15% have almost no annotation on them at all. And this is a logarithmic scale. This blue line shows that there's a proportion of the genome which has an awful lot known about it. And we can show that over time, the rich become richer in terms of protein and gene annotations. You get more and more known about few things, and the effort isn't spread equally. And it could be there's very, very, very useful things in here. We actually show that domains of unknown function, which might have folds, certainly we don't know what the structure is necessarily, but the alpha fold prediction, there's a lot in this unknown fraction. Anyway, we've got this database now. You can find how well known your protein of interest is. So that's a very well known protein with lots of annotation. I think it's the most well known protein in history, beta-catenin, and then this is a very unknown one. But it's it's conserved across eukaryotes, so you must be doing something interesting, I would argue. Okay, going on to some tools you can use for your protein sequences. So just a light overview. Let me know if I've missed your favorite tool or find something else out that's particularly useful. You can do pretty much everything that's simple on the web, although you can install programs locally. I recommend going to these hub sites, and I list them here. So if you want to do something like predict the isoelectric point of a protein, or display an alignment in a pretty way, or do an alignment, or make a phylogenetic tree, or anything you might expect to sort of do on a computer. Check these out, and they sort of classify them into groups, and you can usually find what you want. If you can't find what you want, maybe contact Stephen or I. Maybe we have a better idea, or we can help you write a little script to do things. One of the most common kinds of tool you'll need to use is the protein sequence alignment tool. Pretty much everybody's heard of BLAST and used BLAST. Do be aware, however, that just using the original BLAST, or what's called BLAST Plus now, a slight rewrite, is not necessarily the most sensitive way of using BLAST. There is a much better method called SciBLAST, which is an iterative way of doing searches. So if you're after sensitivity, consider SciBLAST. It's not much slower than normal BLAST. And the idea is that you get some sequences from an initial search, you do an alignment, and then you search again with the alignment, which is more representative of what you're trying to find. Even better, but slower, are HMM-based methods for doing protein sequence alignments. And I am a great fan of HUMA and HH Suite. HUMA actually was initially an LNB product, I do believe, years ago. And it will do a sort of SciBLAST-like iterative thing. But these HMM-based methods, they take a more statistical approach than SciBLAST. SciBLAST just takes an alignment. The HMM-based methods have a statistical model of the probability of one kind of residue following another one. And so it can better store patterns of what is allowed in terms of contextual patterns for a given family. So it offers more sensitivity. When you do these kinds of searches, it often reports something called an e-value, just to make sure people know what an e-value is. It was introduced by BLAST years ago. And this is a bit like a p-value, I guess. But it's the expectation of how many hits you'd expect at random for a given alignment score. Okay, so smaller is better, is kind of the point. And there's a little bit of a caveat here, is that if you're searching with a small protein sequence, peptide, then you're never going to get very good e-values, because the probability of having a random hit goes up quite considerably. Okay, so you need to sort of select e-values based on the length of your protein. Typically, you'd only want to select things with an e-value of 1 in 1,000 or 1 in 10,000 even. But that will suggest that something's homologous, but you might want to go even more strict or looser depending on what the question is you're asking. For multiple alignments, for proteins, I can recommend 2 programs. One is Muscle, and the other is Cluster Omega. I like Muscle because it has extra rules for the hydrophobic cores of proteins, which have different substitution rules. It's still quick. It doesn't go overboard in terms of trying to make everything precise, but it works much better for folded proteins. Cluster Omega I like, which is the evolution of Cluster W, because you have all sorts of options to align alignments to alignments and alignments to sequence and sequence to alignment and all the rest of it. And that's great if you want to get sensitivity out in a SciBLAST kind of style. And this is sort of the strategy I use actually sometimes, sort of doing my own SciBLAST, sort of iterative searches. If you've got something that has a domain that's promiscuous, this can really help. So say if you have a kinase domain in your protein and you do a normal BLAST search and it comes up every single kinase known, then that just sort of muddies things. You don't quite know what to choose, and then you have to search through long lists. So this strategy sort of reduces that and focuses more on orthologs. So the idea is that you do an initial search, and then you select the best thing in every given species, which will generally be the ortholog. Then you do an alignment, this is what SciBLAST does, and then you search again. And you can repeat this cycle. And this will focus on the best in each species only. And you have an alignment that is mostly enriched in orthologs. And you can also filter by length, just make sure things are not truncated or parts of proteins that might be doing other things. Incidentally, I'm writing a web server to do this automatically, but it's not quite ready yet. Okay, you've got a multiple alignment. Maybe you'd like a phylogenetic tree. There's this website here, which pretty much does everything for you. You can have a go at that. So the idea is that you have an alignment, you chop out blocks that are not really well represented in all sequences, for example, where you've got loops or the termini, and it will run the programs that construct trees for you. When it comes to making trees, it's actually can be quite a complicated optimization problem. So you have a whole range of simple through to complicated methods. The most simple is just neighbor joining, which is usually fine for sort of simple questions. The maximum likelihood method are reasonably quick, and they're often a good compromise for speed and accuracy. If you want to go the whole hog and have the most accurate possible, the go-to program is Mr. Bayes, but it is hideously complicated. However, if you want to ask very detailed phylogenetic questions, or you have evolutionary data that's not just putting in sequences, if you've got, say, inheritance of bone shape, features, whatever it is, non-sort of canonical stuff, you can put that into Mr. Bayes. Okay, I can help you with working with that if you want to. Anyhow, this will construct a tree, but then you want the tree in a nice sort of pretty picture. And I love Dendroscope for that. This generated this, which was for Anne Bertolotti, which shows everything in this very pretty form. Okay, so those are my recommendations there. All right, enough about tools. I just want to talk a bit about rigor in bioinformatics, because it's often forgotten. People often do bioinformatics in a panic when they need to write a paper. However, we need to be cautious, and over the years, I've come increasingly to think of bioinformatics as just another kind of experiment that you have to have controls for. Okay, I've had things fail silently until you realize that figure looks wrong when you're just about to publish. So I encourage you always to include controls in your bioinformatic analyses, positives that should always work, negatives that should fail. And just look at the data, plot it, plot scatter plots, plot histograms, and you can see the outliers. Does the shape make sense? I also like to understand my outliers. If there's something unusual, is it something that makes sense? Like here, what's going on here? Well, that's just what happens when you take logarithms. What are these things up here? Oh, these are just P53, etc. I know what they're doing. So you need to understand your distributions, understand your outliers, make sure nothing's gone wrong. There's something went very wrong in this yellow experiment. This was unanalyzable in the end, but there we go. And of course, bioinformatics is mostly just the prediction. It's just the guess. You'll get far more traction in terms of publication impact if you actually go and validate these with some proper experiments, or further experiments, I should say. Okay, in the end, rigor, will be expressed in terms of statistics. Papers are journals are much hotter on requiring statistics these days. I include this slide as a little sort of helper to get you started in a very deep subject. Often you've got some data and you want to make sure that it's real, it's valid, it's not random. Okay, but when you do that, you're going to make sure that you are using the right statistical test. So I've got a little sort of series of questions here, and what these questions do is they point you to the kind of statistical test you might be interested in. Okay, this will be, have to be tuned to your particular problem. So, for example, if you're interested in are two or more distributions the same, have a look what ANOVA is, or Kruskal-Wallis. Okay, this just sort of helps you sort of get into the vocabulary. It can be awfully intimidating approaching statistics, and you have these names of lots of names of dead people, and it doesn't mean very much. Okay, so this is a little hint to go in and find the right one. Now, as a generality, okay, when you're doing a statistical test, each statistical test will assume certain things. Does it assume my data has the same variance on each side? Okay, does it assume that it's a nice normal Gaussian distribution? Know what the assumptions of the test are. If you have something that assumes normality, that might be the best thing to use if your data is normal. So you might want to test normal. Because if you have a test that is, shall we say, non-parametric, that doesn't assume a particular distribution, those are actually weaker tests than having a test that matches the distribution. Okay. Don't forget multiple testing corrections. So you might say, oh, I'm happy with having a threshold to accept a p-value at the 5% level. All that's going to mean if you're testing thousands of genes is that 1 in 20 is going to be wrong, or it's going to come up at random, basically. Now, that might be fine, but don't quote the p-value and select hits based on that. Do a multiple testing correction like we mentioned down here. Yes, know what the test's null hypothesis is, know what it's sort of comparing, what random thing it's comparing against. Sometimes you can get quite different outcomes with different backgrounds or different random situations. For example, if I'm searching for something unusual, like a motif in a protein sequence, do I compare with all proteins, or do I compare with all membrane proteins, or all sequences near my membrane span? The background you choose can make a very big difference to whether you detect something statistically relevant. Okay. So in some sense, you could say, oh, yes, this is statistically interesting, but it's just showing me a typical membranes, near membrane region. It's not actually a signal sequence, for example. When you do these tests, remember that particularly when you're setting thresholds, stringencies, am I interested in sensitivity or reliability? Do I want to find as much as possible and so accept false positives, or do I want it to be totally reliable and have no false positives? You're going to have to make these decisions yourself. There we go. Okay, so that's in there as a bit of a guide. Okay, so as a light thing to sort of end with, and move to machine learning. Next year, it is planned, I've just told Stephen it's planned to have a machine learning lecture in this series. I think he's happy with that. Now, machine learning, or AI, is all the rage these days, but it's, if we ignore the AI that's trying to pretend to be a human, it's nonetheless a very useful tool to biologists. And the idea with machine learning is that if we can use statistics or some sensible mathematical route to answer a question, use that instead. It's robust, it's reliable, you can explain it. Machine learning is when there's data, there must be some pattern, but no obvious kind of connection between them. Okay, how would you model translating English into French? It's not obvious, it's too complicated, or the protein, or guess phenotype from polymorphism, whatever it is where the link must be there, but it's not obvious, it's complicated. Be aware, though, that machine learning can only be used if you've got good quality and abundant training data. You've got to have, it learns by looking through lots of examples. You must have a lot of data. Okay. And we use it especially, as I mentioned, where there's complicated problems, which means there are lots of degrees of freedom in the data, where you've got high dimensional data. So this means that, like a protein sequence, you have lots of different positions in my sequence and lots of variations within that. This is a big vector to describe it. You need lots of numbers just to describe one data point. That's what we mean by dimensionality. Machine learning is also particularly useful when you have mixed kinds of data. In fact, it doesn't really care about the source of the data as long as it is predictive. So, for example, I'm trying to predict protein complexes from mixing RNA-seq data with proteomic data with phylogenetic data. Machine learning doesn't care. It learns, it weights it appropriately so that it can come up with the best machine that makes the best prediction. Machine learning also is very handy when you've got missing or incomplete data. A lot of statistical methods just fall down because you're missing something, or there's a zero or not a number there. Machine learning generally doesn't care that much and can be trained to ignore missing data. In fact, that's what we do typically. We typically train with a neural network with things blanked out randomly, and it has to learn to cope with that. So it can be an ideal choice if there's missing data. Machine learning does sound complicated when you have somebody like Demis describe how wonderful it is and how intelligent he is, but in its fundamental sort of roots, it's actually quite a simple idea. The simplest machine learning method, I think, is this idea of the k nearest neighbors. And what you do is you say, well, I've got some data and I'm going to represent it as a vector. Imagine the example I give is like colors, red, green, and blue. Those are three numbers. They define a vector, a position in color space. You have to have sequence space, whatever. And you've got some data, some vectors with a classification. You have some space, you plot them in, and the idea is that you can classify a new data point by getting its vector and seeing how close annotations are to its vector with the unknown classification. So here my query with a question mark is close to vectors in class A, class B. So you know it's not class C and it's probably class B. K nearest neighbor just takes a pole within a given radius in Euclidean space. So that's as simple as machine learning gets. And in some sense, the deep neural networks are not doing anything much more complicated than that. They just do lots of it in multiple layers. So machine learning, you can think of it as being a bit like clustering and discrimination methods, like PCA. And we talk about machine learning models, but a model, in some sense, you can think of it being like, one model is having clusters with boundaries, and then where the new thing goes is, you know, inside one of those boundaries, and that makes a prediction. That's mostly what machine learning does. Or it can do regression, where it has a smooth sort of continuation between categories rather than just a hard boundary. When it comes to machine learning, if it's a fairly sort of straightforward problem, and the data is not immense like a protein structure or natural language, have a think about whether you could use one of the canonical machine learning methods like support vector machines or random forest. Don't necessarily dive into neural networks. The reason being that SVMs and random forests, they've been used for many years, they're robust, they are quick, and they have explainability. You can know why very easily certain decisions were made on certain data points. Okay, and Python has this nice scikit-learn library where you can just use these canonical non-neural network and simple neural network-based methods. And you can actually visualize, in this case, how they're making their decisions. You can see all they're really doing is they're sort of dividing boundaries between data classes. Deep learning is often unnecessary. Okay, but sooner or later you may need a neural network, and I will be offering Python-based courses on neural networks. It used to be in the olden days that neural networks were difficult because you had to code everything yourself from scratch, pretty much. These days, we've got some lovely libraries in languages like Python and Julia and R, etc. Most popular of which are PyTorch first and then TensorFlow. PyTorch is the best for getting started, I would say. TensorFlow is the more industrial one. And people deploy industrially with that more. But the idea is that all the sort of mathematical operations you need to do for neural networks, which is just a lot of matrix multiplication really, is taken care of for you pretty easily. I mean, here's a neural network written in sort of 10 or 11 lines of code. Okay, so the libraries allow you to take input, you define the layers. This is what your neural network's doing. It's sort of here. It's dropping out. It's sort of nulling 20% of the data. It has not going into neural networks, but it has sort of 3 layers, and then it spits an output out. You can write very simple code to do quite powerful things these days. It's all taken care of for you. My old courses are available here. They're for TensorFlow. You can look at those. Okay, so yes, it can get very complicated, but start simple. And to help you along with this, Stephen and I and some others offer courses. This is our new website, which will be advertised soon once we've had a little test of it. It works, but I need to test all the edge cases. And Stephen runs courses on command line computing, so running stuff on your Mac and on the cluster, and some long detailed Python for data analysis courses. I run some machine learning courses, neural networks, general machine learning. Nick's crew have actually run some microscopy courses through our system in the past as well, so on scripting and image analysis. I also run some very simple Python starter courses as well. So look, this will be advertised when it's ready, so look out for that. And then we will continually have little courses on there on a rolling basis as well. And if you've got any suggestions for courses you'd like to see, we're going to send out a survey soon, so make sure you fill that in. Okay. Thank you for your attention. Almost on time. Any questions to Stephen or myself? Send us an email if you think of something later, but yes, hi. Yeah, so this is Stephen and I. So I was in Sean Munro's lab, And we started a project where, this is a follow-on from the unknown project. We want to know what proteins do and how to automatically mine information. And what we decided is that we'd like to look at lots of different kinds of experimental data all together. Hence, we're using neural networks. So we look at expression profiles, protein abundance profiles, phylogenetic profiles, and also subcellular localization, and something Stephen mentioned, which is depth map, which is essentiality profiles. And what I mean by a profile is a protein's abundance or essentiality or whatever it is in different tissue types. And the point being that proteins that are in the same complex or the same pathway will tend to be in the same place at the same time, or at least surprisingly often. So, you take, two things that are in a complex, which is the most strong association we can think of. Their abundances will correlate. Forget stoichiometry, but they will be expressed and be almost equally abundant in the same tissues at the same time. And so that can be a fingerprint. So we've trained a neural network, and there's actually a website you can try now. We haven't published it, but you can search it and see if we've picked up any interactions. We are picking up a lot of the known things that haven't been annotated properly, which is pleasing, but also hopefully there'll be some new things in there. The trouble is, it's very difficult to set the difference between sensitivity and reliability. So that's what we're working on at the moment. But it doesn't give too many false positives, but it gives just enough false positives that shows new things, I think. So if you're interested, email me and I will send you the link and you can just try it now. One of the things I haven't mentioned is that as part of a wider initiative to connect bioinformaticians together within the building, I am going to start a new website, which is going to be very similar to that one. It's not going to be the same site, the same idea, where we get all the bioinformaticians who are in the whole building with their expertise areas to put themselves on the website and advertise all their slides and websites and just their general expertise together. So we'll have a centralized informatics resource, bioinformatics resource, so nothing to do with computing.
And we've also set up a Slack channel.
Oh yes, there's a Slack
days ago on the LMB.
So if you want to be bothered with chit chat,
the old memes about bioinformatics and things like that.
So yeah, the bioinformatics community was starting to organise it somewhat better than, because we're not a service, but we are a collective. Resistance is futile. So.